All right, good afternoon. I'm just going to sit down and record the last couple of slides to start to finish off Chapter 7. And remember, after this, there's an old lecture from 2022, but it happened right after a Nobel Prize was given that ended up being exactly about sugars and how they work. And I want to show you that. And so I'm going to bring that back. That'll be 15 minutes. Hopefully this will be 15 minutes or less. So let's get to it. All right, so we ended up with this. A proteoglycan has a glycan is the most of it. So chondroitin sulfate is one of those interesting ones. And so the thing about the sugars that end up on the important ones of these, they are often glycosaminoglycans. They're called GAGs for short. And they end up with there's a lot of aminos in them for some reason. I'm not sure we even know exactly why, but it's something that we've noticed. So chondroitin sulfate would probably be one of these. The end result is that you have a peptide that's extremely negatively charged. The most negatively charged biological macromolecule known to science is heparin. And heparin is also important. You may have heard of it somewhere. I would ask you if you've heard of it, but if you've watched a TV show, they're looking for heparin in an emergency room. They say stuff like, I need some heparin stat. And the reason is heparin is an anticoagulant. It's natural. You can make it in one species, and it's pretty much the same as in another species, so you can purify it out. I don't know which species are the best, but I do know it's possible. Because basically it's not doing specific functions. What it's doing is sort of making the blood more gel-like. It's just spreading out in the blood. And it's sort of making it so that clotting is proteins coming together in the blood to make a clot protein. The proteins can't get to each other as easily if there's a lot of heparin around. The gel-like nature of the blood keeps them from coagulating, keeps them from touching, keeps them from clotting. And so heparin is actually made by specialized cells. It's like the whole molecule. And a lot of it is based on heparan sulfate. So you might see heparan sulfate. We usually use those pretty interchangeably just because it's easier to say heparan. But you should understand that heparin has this structure. So here it is, glycosaminoglycan. And it's going to have, you see it's got the aminos there, it's got the sulfates there, it's got alpha linkages, so it sort of spreads out as a big helix. It's not a sheet that's really packed together, it's more of a looping structure. And it's got some weird sugars in it. I'm not expecting you to memorize this, but I want you to count the negative charges. There's 2 on one of them, there's 3 on another one of them. Basically, this is why it's the most highly negatively charged molecule known to science, because each sugar has 2 or 3 negative charges on it, sulfates, or in the case of the one subunit, an acid group. So, we can actually look at glycosaminoglycans using this STM scanning tunneling microscopy. So here's the structure is all the way on the right, and you see this has glucnac and the acid form of glucose, glucuronate. And it has alternating forms of that. You can see the structure on the right goes with the diagram on the left, and then you also have, I think I'm backwards, so I should be doing this, and then you have in the middle what they actually saw. And you can see on this, they have the version without sulfates, and then they have the version with sulfates. And if you look closely at this, the version without sulfates is more curved. The sulfates are repelling, they are straightening out the structure, and even though it's a mixture of beta and alpha linkages, it should be not exactly straight. The sulfates make it more straight. Be able to look at this, I would like to be able to give you this figure and basically have you repeat what I just told you. So if you can have these, the other thing they looked at in the paper is these glycopeptides, and this makes perfect sense. If you have an OH group on an amino acid, you can do the same chemistry with that. And it turns out that sometimes when you have an amide, you can actually do the chemistry that's exactly the same, except that the bridging atom is a nitrogen rather than an oxygen. See, that's happening right here with an asparagine. So you can have glycopeptides where you have covalent bonds, and it's the same type of chemistry. It's just an N-linked glycosyl bond, because glycosidic bond. All the glycs are hard to say sometimes. But here's an example where you have to know your amino acids and you have to know your sugars to be able to look at the 3 ways of looking at the same structure here. Technically 4 ways. So the thing is, you can look at these things, you can talk about the structure, but you need to be able to have the image in your head of what this structure can do. And so heparin, what's a good heparin binding protein? It will have a lot of lysines. In fact, you could even make a polylysine molecule, just a bunch of lysines, and you throw that into someone's blood who's got a lot of heparin. Maybe you want to turn the heparin off. So lysine will actually bind the heparin. It's showing it binding through its sulfates right here. All those red oxygens are binding to the blue nitrogens on the lysine. So all this involves a lot of vocabulary words that we've talked about. At its heart, it's a simple plus-minus interaction. And it's actually a therapy. You can make a heparin blocker just by making a molecule with a bunch of lysines in a row. So if you can do that, if you have the lysines in a protein, if it has a big blue surface, positively charged nitrogen-rich electrostatic surface, the heparin can actually bind to that. So another negatively charged molecule will bind to this positively charged surface, and this is actually a protein that binds heparin, and we can see exactly how it works. This is actually 2 proteins. It's a protein growth factor and its receptor. Both of them have blue surfaces, and you can even look for the blue surface and say, oh, that's probably where it binds heparin. Here's another case. This is a heparan sulfate epimerase, so it's actually going to do some chemistry on the heparan sulfate. So it has an active site. But the question here is not where is the active site. The active site is related to the calcium. And by the way, this is related to your worksheet right here. This is how calcium works to help bind this glucose. That's related to the worksheet that we handed out today. But the question I want to ask is, can you tell on this electrostatic surface up here in the corner, where is your heparan sulfate going to bind? All those sulfates are going to be red oxygens. They will bind to blue nitrogens, and you can even see sort of a groove where that curly heparan sulfate will be able to just bind to that groove, and its active site will be able to reach over and epimerize something in that heparin. So the last thing to talk about is glycoproteins. And a glycoprotein is more protein than with a little bit of sugar on it. Okay? So, and they can for example, look at this. You can see the little bits of sugar sort of hanging off to the side. It looks like Dilbert's boss has hair kind of like this. He has just 2 little tufts of hair that go up. I hope you all still know Dilbert, even though it's kind of an old reference now. But look at it. It looks exactly the same. So it's like the sugar is like little bits of hair. So a bald protein with just little bits of sugar hair would definitely be a glycoprotein, not a proteoglycan. You definitely have the proportions on the side of protein being prevalent here. So the thing is, if you see a glycosylated protein like this, where the sugars have been attached to an amino acid on the protein, it's probably going to be in the extracellular matrix. It's probably going to be either on a cell on the outside or floating around on the outside. Because look outside the cell, and most proteins are going to have a couple of sugars on them at least. Some of them are highly glycosylated. They're basically sugar balls. And they just have the one part that needs to be open to solution open, and the rest of it's covered up. Because, like, you know, the immune system has a hard time grabbing onto carbohydrates. So you sort of protect yourself by putting this sugar shield on it. It's a very flexible structure. It doesn't have many things to grab onto specifically. It has lots of hydrogen bonds, and water can interact with those easily. But it's hard to make an antibody that will bind to sugar. And so then you have, these will protect the proteins from being recognized by the antibodies. So here's the exact showing you what the oligosaccharides look like when they're attached to the proteins. You can have a serine that's O-linked. Many of them are O-linked. You can also have asparagine can link through its nitrogen and is therefore an N-linked sugar. Both of these are different sugars, but it's just showing you the anomeric carbon can attach. It can just do the regular old hemiacetal to acetal chemistry. And why do proteins do this? Well, it's because having sugars around will bound to the protein will help stabilize it. Think about it. The sugar, there's nothing better at interacting with water than a sugar. Proteins have hydrophobic bits. If you cover up the protein, not only do you protect it from like antibodies and stuff like that, you also protect it from pretty much anything, as long as you just put the sugars where you don't want it to react. So it turns out that life figured this out very early. There's a archaeal protein. Archaea are even a different kingdom, but they use the same amino acids. They use O-linked modifications of sugars, and it turns out if you look at their external proteins, they are highly glycosylated. There's hardly any space to see the protein. If you look on the right here, you have hardly any space to see the protein anymore. It's all covered with sugars. They got a lot of threonines, a lot of serines, a lot of and other threonines, and they just see sugars on all of those. This lives at pH 3 and 80 degrees Celsius, and it's got to be some of the reason why it can do that is because right here it has more sugars on it than anything else that we've seen. The sugars help stabilize it. So of course, after you have the initial linkage, the rest of the non-reducing end of that sugar is going to be able to participate in this acetyl chemistry, and you'll be able to grow it, you'll be able to branch it. You can have O-linked or N-linked oligosaccharides that are part of glycoproteins. And again, you can see, the one thing I want you to notice here, notice that for all of these, there's a lot of them that end with sialic acid. Sialic acid is a common cap to an oligosaccharide, and that means that if there is something that will bind to a sugar, it will often bind sialic acid because it has a very distinctive structure. So it's kind of like the fingertips of these things are the purple sialic acids, and that's a real tendency in life. So just to look at how many things, no wonder that we looked at proteins first. Sugars are just so flexible. They sometimes aren't doing anything. They're just blocking stuff. And they have all these different units, all these different linkages. It was just really hard to figure out. Yet when somebody did figure it out, they won a Nobel Prize for it. And I will let you find out about Carolyn Bertozzi in the follow-up video. I just want to show you that there's an oligosaccharide that was one of the main things that they looked at with this STM imaging single glycans paper. And this really shows you vividly the three ways to show the structure: the diagram, the projection structure that you can draw, and then on the right they have the experimental evidence where you can fit that onto the structure, and they're able to actually see these subunits and how they curl. It's pretty cool. You can look at the pink alpha 1 to 2 linkages and try to see if there's differences between those and the black alpha 1 to 6s or the gold alpha 1 to 3s. I think in the paper there's something about that. But once you can find your way around the structure, then you can ask questions about it. One last story. Glycoproteins are really important to cell-cell interactions, and so they're often important to disease. If you have a protein that binds a sugar, it's not easy, but it's possible. And there are some proteins that do specifically bind sugars. We call those lectins. And for example, ulcers are caused by a lectin interaction. It's actually caused by a bacterium. And when I was growing up, they told us that ulcers were just caused by stress. And no, the stress opens a door for the bacterium to come in and infect you. They did not know that when I was growing up because of another Nobel Prize. That is our last story for today. So there's a lot of ulcers that are caused by H. pylori, which is the bacterium that we now know causes them. And it uses a lectin to bind a particular sugar on the stomach lining. The oligosaccharide is called Le super V. It's associated with O-type blood is the main thing. And this is the thing that they should have realized. People with type O blood get ulcers a lot more times than others, and this is one of the clues that it might be caused by a bacterium rather than a general stressor like stress. Why would people with type O blood be more stressed because they're universal donors or something? I'm really not sure. And so if you look at here's the structure. Once they found out that H. pylori was the infectious agent, then they could do microbiology on it. And today ulcers are actually not nearly as much of a problem as they once were because we know what causes them. So everyone used to think that they were caused by stress. But then there were Australian researchers that looked at these clues, Robin Moran and Barry Marshall, here they are, and they actually thought that they could look for a bacterium. They looked for one and they found one. And they said that we think this bacterium is causing ulcers. So how do you prove it? Well, the younger one of them, Barry Marshall, I don't know if seniority played into this decision, he drank a vial of the isolated bacterium and he gave himself an ulcer. He said, great, my theory is partially proved. I want to really prove it by drinking an antibiotic and taking away the ulcer. And that happened. So instead of going through IRBs, he experimented on himself, which I don't recommend it, but it's better than experimenting on others, I suppose. You're responsible for your own side effects. Because of this, because they proved they were right and they changed our understanding of how ulcers happen, they won the Nobel Prize for this. And now this story is proved right. I don't know how many experiments were done by other people who were wrong and ended up, like, I don't know, hopefully not killing themselves or causing bad harm to themselves. But when it works out, it makes for a good story and it does advance our scientific knowledge. So that's what they did. And in fact, if you're infected by H. pylori, it's actually, is one of the reasons why stomach cancer is not as big of a deal as it used to be. Even if you don't have symptoms, being infected by H. pylori makes you more prone to stomach cancer. Infections do have a linkage to cancer. So this is not only an antimicrobial, anti-ulcer finding, it's also an anti-cancer finding. H. pylori is the most cancer-causing pathogen we know. If you're curious about that, check out this paper, but that's cancer biology, and this class is done. So we're going to have one more worksheet on the review day. Other than that, you've got everything you need for this class. Please be preparing to discuss the paper. We're going to be working on that in class on Friday. And don't forget, we have one more old lecture from 2022 to have 3 stories that will follow up on these, including bulletproof wood. All right, so I'll leave you to go watch those now.